Hi there, my name is Callie Chappell. I am a PhD candidate in the Department of Biology at Stanford University, and this is a talk that I gave for the American Society of Naturalists meeting in Asilomar, California in January of 2023. The session is about competition, and today I'll be sharing with you a talk about eco-evolutionary drivers of priority effects in community assembly. Now, here in Asilomar, which is in Monterey, California, we have a lot of amazing tide pools. In fact, when you walk outside this conference center, many people here have gone tide pooling, and the marine coastal ecosystems are perfect examples of where people have studied how communities change over time, a process called community assembly. And one classic example are these kelp forests that represent alternative community states, where you can have kelp-dominated communities, but with some perturbations, they can actually move into different community states, alternative community states that are, for example, urchin dominated. Now, in this talk, I'm not going to be telling you about kelp. I'm actually going to be sharing a different system, but the questions are the same. What drive alternative community states, and what are their functional implications? These are two questions that I hope to answer in this talk about Diplochus or Mimulus aurantiacus, the sticky or bush monkey flower. These are a native California shrub that are broadly distributed and are the focus of our research because they have microbes, bacteria, and yeast that live in the nectar of every flower. So each flower represents a discrete ecological community, and there's dispersal between flowers by pollinators such as Anna's hummingbirds pictured here. Now, I'm going to tell you about a field survey where we studied these nectar microbes, but first I want to acknowledge that this fieldwork was conducted on land stolen from the Esselen tribe of Monterey County, the Mucma Ohlone tribe, the Amamutsan tribal band, the Ramaytush Ohlone, the Tamian Nation, and the Coast Miwok. The research conducted at these field sites benefits from Indigenous stewardship, and our lab, Stanford University, and every institution in the United States has benefited from, historically and to this day, Indigenous peoples. So I want to acknowledge that contribution through this fieldwork, as well as the many fundamental ideas that have guided this research and ecology and evolution as a field. So as we look across this landscape, we thought about asking how distributions of nectar inhabiting microbe varied across this coastline, going all the way from Bodega Bay up here, all the way down to Big Sur. And we are here at the tip of the Monterey Peninsula. So in fieldwork led by Manpreet Dhami and a lot of intrepid undergraduate, graduate students, postdocs, and faculty, they went to all of these field sites and collected sticky monkey flowers, over 1,000 of them. And they measured bacteria and yeast in each flower. So you can see some examples of how we picked flowers. This is actually associated with class, and then cultured them on agar plates. And this is what we found. So each one of these points represents a single flower and its microbial community. And you can see that these blue points represent a variety of bacteria-dominated flowers. So these flowers had a lot of bacteria, but just that. And these flowers highlighted in yellow were yeast-dominated flowers, so flowers that had a lot of yeast. And you might notice that flowers seem to have either bacteria or yeast, but very few had both bacteria and yeast. So this suggests that we see alternative community states in these nectar-inhabiting microbes. And prior work in our lab suggested that one mechanism that might lead to these alternative states are priority effects, where, for example, yeast might arrive first to a flower, change the environment, for example, use up the amino acids or utilize the resources such that later arriving microbes aren't able to grow, and that flower ultimately becomes dominated by yeast, whereas the opposite might be true with bacteria. Now, in this talk, I'm going to be focusing on the priority effects of early arriving bacteria on yeast, but we've done a variety of work looking at priority effects focused on the yeast-mediated priority effects, which I'm not going to talk about in this talk, but you can check out some of these papers here if you'd like more information. Now, to study this, we suggested that this might be true in the field, and prior work focusing on yeast-yeast interaction suggested that priority effects might be at play, but we actually hadn't done any experiments to test this directly between bacteria and yeast, the key interaction based on our field survey that seems to be leading to these alternative states in the field. And so two amazing undergrads, Veronica and Katie, decided to do a lab experiment in order to test this. So instead of using actual flowers, they used tubes with fake nectar, and they did an experiment where they inoculated either bacteria or yeast into these tubes, let them grow. A couple days later, added their competitor. So for example, if bacteria arrived first, they would add yeast, and then after 5 days, about the flowering period of our plant, they measured how well each of them grew. And again, I'm just going to focus on the effect of bacteria on yeast here, but they also did the reciprocal experiment. And this is what they found. They found that when yeast came first, they were able to grow pretty well, but when bacteria came before them, their growth was depressed, suggesting that bacteria exert a negative priority effect against yeast. Now, these undergrads wanted to know, well, why is that? What are the bacteria doing to prevent yeast from growing when the bacteria arrive first? And they actually were able to take inspiration from some other undergrads, Juhi and David, who were involved in this nectar microbe research almost 10 years before them. They were participating in Bio 47, or at that time known as Bio 44Y, a course-based undergraduate research experience. It's the intro to ecology and evolution lab at Stanford. And instead of doing kind of textbook cookie cutter labs, they actually participate in original research studying nectar microbes in the field. And when they were doing this class, they wondered, what about nectar pH? And so Katie and Veronica in their lab experiment actually measured how bacteria influence nectar pH, and they found that as bacteria grew to high densities, they actually dropped the pH of the nectar to around 2, the pH of lemon juice. That was really striking, and we might imagine that yeast might not be able to grow very well in pH equals 2. But then we wondered, okay, that's very interesting in the lab. Does that hold up in the field? And so just last summer, two amazing undergrads, Yadira and Fatu, I'm going to say every undergrad's amazing because they are all amazing. They did a field experiment where they went into the field and they measured the nectar pH of each flower and also cultured the bacteria. And they found the same trend, that as you had higher densities of bacteria, they dropped the nectar pH truly to around 2, which was very, very low. So this suggested that there's these alternative states between bacteria and yeast, that they are likely because of priority effects, and the mechanism of priority effects between that bacteria exert on yeast is likely dropping pH. So does this hold up? Does this mechanism of pH actually hold up in the field? And we did another field experiment where we sampled many flowers that was supported by Lexi Golden, an undergrad, and another amazing undergrad, and Mark Bitter, an amazing postdoc, and Tad. This is actually a picture of Tad's hand, where we went out, picked a lot of flowers, and measured their pH on pH strips. And this is the distribution of pH that we saw in flower nectar from the field survey. So we noticed that there's trimodal, and this first mode, we seem to think, again, we didn't actually measure the microbes here, but we thought maybe it was fresh nectar. So these high pH flowers had fresh nectar. These middle pH flowers might be ones that had yeast growing in them. This is a hypothesis again. And then these low pH flowers might be the ones that had bacteria. But these were just hypotheses. But then we remembered that a former postdoc in the lab, Rachel Vanette, actually did a lab experiment. And so what Rachel did was she went out, she took sticky monkey flowers from the field, she took their nectar, filtered it, and then inoculated different microbes like bacteria and yeast into it, and then measured how those microbes change the nectar pH. And what she found in the lab, I'm going to show on top of that field data. These are these dotted lines. What she found in the lab is that it matched almost exactly to our field observations, that fresh nectar without any microbes had a pH of like slightly below 8. That nectar that she grew the yeast in was around 5.5, and the nectar with the bacteria grown in it was around 2. So this really supported our field observations and our predictive mechanism to what might lead to alternative states and the mechanism by which that happened. So what we found was that there's these alternative community states between bacteria and yeast, and the mechanism likely is that by bacteria lowering nectar pH. But what about that second question? What are the functional implications? If we know the mechanism, can we use that to predict why certain the effects of these alternative community states in real environments? So I'm going to tell you two stories, one about evolution and one about ecology. So starting with evolution, getting back to our field survey, we noticed that across all of these different field sites, they varied in the proportion of flowers that were dominated by bacteria, for example, versus yeast. So for example, this site, Site 2, Muir Woods, had a lot of flowers that had bacteria that were dominated by bacteria, whereas this site, San Gregorio, down here, mostly had flowers that were dominated by yeast. So could the fact that the relative proportions of bacteria and yeast differed between sites drive rapid evolution or different selective pressures within each of these sites in a meta-community? So to test this, we wanted to know whether we could evolve yeast to resist priority effects. So we took yeast that we isolated from the fields that I'm going to call ancestral yeast, and we serially passaged it in 2 different nectar types. And the way that we did this was by growing them in fake nectar, and then every other day taking 1 out of 10, so 1/10 of that volume, and transferring it into new environments. This is a really common experimental approach in experimental evolution, but it actually has really nice natural history parallels in our system because it's just like those yeast moving every couple days from new flower to new flower on the beaks of hummingbirds. And we did this in 2 different environments. One are just normal nectar that we've been doing our experiments in that represent a neutral environment, and one a low pH nectar that represent this hypothesized mechanism of priority effects. So at the end of about 2 months of doing this, which represents the flowering season of our plants, we had 2 different types of evolved yeast. We had yeast evolved in the normal nectar and yeast evolved in the low pH nectar. And you see 4 tubes here because we did this with 4 independent evolutionary trajectories, or 4 evolutionary replicates. So what we could do with these evolved yeast, as well as the ancestral, is compete them against bacteria like we did in those experiments I told you about before, and ask how susceptible are they to priority effects. So here on the y-axis is how susceptible they were to priority effects. And we found that the yeast evolved in the ancestral yeast and the yeast evolved in the normal environment were quite susceptible to priority effects by bacteria. They were pretty negatively affected by bacteria. But the yeast we evolved in the low pH nectar were much less affected by bacteria. They were resisting the priority effect better. This suggests that pH seems to be a mechanism that's recapitulated through evolution to resist priority effects, supporting our hypothesis that this is the mechanism. And this research was conducted with an amazing team of undergrads called Team Nectar Microbe. And the other undergrads were also part of Team Nectar Microbe, but this was part of a really big joint effort with Clara, Addie, Brianna, 2 Jonathans, and Paloma, as well as some other students who were part of Team Nectar Microbe who weren't involved in this particular study. And I'm really proud to say that they worked not under me, but alongside me. Together, we came up with the hypotheses, we designed the experiments, we did the analysis, and we thought of future experiments as a collective. We had our own lab meetings, we had our own lab values, we even did service projects together, one of which was writing a post for the blog Dynamic Ecology about mentorship in the time of COVID-19. You can use this QR code to check it out. So I just want to highlight that this work, all of the work was done in a huge collective team effort that really celebrated kind of diverse perspectives and experiences that we all have that make the science really good. And one question they had was, well, this is all fine and dandy in the lab, but what happens in the field? So they did more experiments where they actually compared the diversity that we see in the lab to that in the field. And so we took that same strain of ancestral, we're going to call it MR1, that's the name of the strain, and we did the same experiments with 2 other strains that we isolated from the fields. And we found that those strains, MR1 was most susceptible to priority effects in both the field and the comparison to the lab evolved and the field evolved. But these 2 other field evolved strains, or just strains that we isolated from the fields, they were less affected by priority effects to a similar extent, actually, as the lab evolved strains. So what this suggests is that we were able to recapitulate phenotypic diversity that we see in the field in the lab just by knowing, or this hypothesized mechanism of priority effects lowering the pH. Very cool. But then they asked, okay, well, that's all fine and dandy, but if these guys are so good at resisting priority effects by bacteria, why is this one that's bad at growing with bacteria still around? Why is this one still in the environment? And fortunately, our lab experiments actually could give us some insights into that. We found a trade-off between resistance to priority effects and growth alone. So if you're really good at resisting priority effects by bacteria, you're probably not able to grow that great, and vice versa. So this resistance to priority effects actually trades off with growth. And this provides empirical evidence to support a purely theoretical hypothesis that we had that was published actually in AmNat in 2018 by former postdoc associate and Tad in our lab, called eco-evolutionary buffering. And so getting back to our example before, here at Muir Woods, remember that was a site that had a lot of bacteria-dominated flowers. So maybe at that site, there was selection for resistance to priority effects because there were a lot of bacteria around. Whereas at this site, maybe at the San Gregorio site, where there weren't very many bacteria, it was mostly yeast, there might be more selection for just being able to grow real fast. So this eco-evolutionary buffering could be a mechanism to maintain regional coexistence because you see heterogeneity between sites in selective pressures that then could support regional coexistence of, for example, strains that are good at growing alone and strains that are good at resisting priority effects. So to summarize, we found an evolutionary implication of these alternative community states where rapid evolution can promote regional coexistence. And now I'm going to tell you about an ecological interaction related to plants and pollinators. So the students in the Bio 47 class I was telling you about, that undergraduate lab class, they did an experiment over several years where they actually made fake flowers out of wrapping lab tape around PCR tubes that they put different nectar types in, because they wanted to know, okay, what does the difference in nectar chemistry, how does that influence pollinators? And you could see from this video that the pollinators love coming to these stakes. Here's a picture by Maya Zhu, an undergrad who taught me how to do bird photography. I'm still not very good at it, but hopefully I'll get as good as Maya is one day. And this is what they found. They found that flowers with low pH nectar, so that like lemon juice-like nectar, pollinators drank a lot less of it than the normal nectar. So this suggests that pollinators dislike low pH nectar, and this supports prior work in the lab that showed that when flowers in the field were inoculated with nectar bacteria, they had lower seed set. So this could be a mechanism by which the low pH, so one alternative community state, the bacteria-dominated communities, alter plant fitness. So another functional implication here is that low pH can alter plant fitness by changing pollinator behavior. So to summarize what I told you about today, we found that there's alternative community states in these nectar-inhabiting microbes between yeast and bacteria. The mechanism by which bacteria exert negative priority effects against yeast seems to be by lowering the pH, and there's evolutionary consequences related to eco-evolutionary buffering and potentially regional coexistence, and ecological consequences with plant-pollinator interactions and plant fitness. There's a huge number of people to thank in this project: amazing undergraduate team, the Fukami lab, all the people who were involved in the original survey, experimental evolution folks. There's a big section on genomics where we did whole genome resequencing of the evolved ancestral lines to look at potential basis for evolution to priority effects that I didn't talk about in this talk. And of course, the amazing team of instructors and staff associated with Bio 47, as well as our funding through the Joint Genome Institute and the National Science Foundation. The results that I showed in this talk are all part of a paper that we recently published in eLife entitled Wide-Ranging Consequences of Priority Effects Governed by an Overarching Factor. You can take a look at that talk in the video description here, as well as this QR code. And something I want to highlight that we're really proud of about this paper is the diversity of co-authors, which include undergraduates, grad students, postdocs, faculty, but also staff from the Bio 47 class. We also have a papercast version of this paper where you can actually listen to it, and that I'm reading it if you like the sound of my voice. And I also include verbal descriptions of all of the figures. And we really believe that the diversity of the people who are involved in this project contributed to it, what we believe is great science. And so we also have translated versions of the abstract into 5 languages, representing the first languages of all of our co-authors. So thank you so much for listening to this talk. And I'll just leave this slide up here that this talk was given in Monterey, and Monterey is part of the unceded land of the Asilomar tribe of Monterey County. The Asilomar tribe is fantastic, and I highly suggest that if you've enjoyed this conference, to donate to them. This is a QR code that will take you to their website. And I just want to leave you with a win that they were able to rematriate around 1,000 acres of land in Big Sur in 2020. And this is a picture of that land. So just want to end with saying, as ecologists and evolutionary biologists, we should support the rematuration of land to Indigenous people as a central part of our praxis in caring for and about the environment and all of our human inhabitants. Thank you so much.